What is polypeptide synthesis? The biological process known as polypeptide synthesis produces peptides, which are chemical molecules made up of peptide bonds connecting many amino acids. They are fundamental units of protein which are necessary for the proper operation of cells. As the fundamental components of proteins, amino acids as the nitrogenous scaffold for molecules such as hormones, as stated in the article of ArtSmart. A protein such as insulation or alanine is made up of one or more polypeptide chains that twist and combine to form a three-dimensional protein. The transfer of genetic data from DNA to RNA and the synthesis of protein, likewise referred to the central dogma. The central dogma of molecules biology describes how genetic information moves from DNA to RNA and into proteins, which are functional products. This can include the function, structure, regulation of the protein according to the core diagram. RNA is the messenger that transports the information from DNA to the ribosomes, where it's used to synthesize all of our proteins. Polypeptide synthesis occurs in two main steps, transcription and translation. Within the first step, transcription occurs in the nucleus, which is the location of the DNA. The genetic code is what gives an organism its physical characteristics and psychological functions. Segments of the DNA are converted into mRNA molecules during transcription. RNA is the smallest single-stranded composition of ribosomes and has the capacity to exit the nucleus through nuclear pores as compared to DNA. The transcribed copy of the permanent instruction from DNA nucleotide sequence is carried by the mRNA during polypeptide synthesis and enters the cytoplasm where ribosomes are found and protein synthesis occurs. The bases arginine and thymine are complementary base pairs, while cytosine and guanine are complementary base pairs. However, with RNA, the nitrogen base of thymine is replaced with uracil. There are three phases of transcription: installation, ligation, and termination. Starting with installation, the RNA polymerase enzymes indicates which base should be transcribed during installation process, then attaches itself to a promoter. A specific DNA sequence that is instantly discovered. This allows the hydrogen bonds between the nitrogenous bases of both strands to dissolve, causing the segment of DNA, a double helix, to unwind. As a result, two complementary DNA split apart, supplying the single-strand template required to create the mRNA strand. In the second stage of transcription, ligation takes place. Here, RNA polycaterizes the synthesis from RNA to utilize the central strand as a molecule to create the complementary strand of mRNA that presented in the cell nucleus. At the start codon, RNA polymerase starts the production of mRNA. It then proceeds through the gene, reading the sense strand of both 5 and 3 directions. The nucleotide chain is processed, travels to the 5 and 3, and ends up in the full RNA nucleotide and the end of the mRNA case polymerase. RNA polymerase released the RNA chain to the complex transition comes to an end. The mRNA is now ready to indicate translation. Nevertheless, the mRNA strand is considered pre-mRNA and needs additional editing before it can leave the nucleus and direct translation. This kind of editing is called splicing. Pre-RNA is considered composed of exons or coding sections and introns or non-coding regions. During splicing, a molecule known as small nuclear RNA or SNOBs joins forces with protein to generate a small nuclear proteins. These SNOBs bind towards the end of the exon before ligate to form splicing zone. When an exon forms, the splicing clears the neutron and the intron is released into the nucleus by the enzyme known as exon nucleate, where it grades. By removing the exons, the remaining codon sequence is a protein synthesis become an entire instructions. Following splicing, the poly A is added to the 3 and the module added to the 5 with pre-RNA, both of which shield the mRNA being collapsed on the inside. After splicing has occurred, the pre-RNA is considered to be mature RNA and is ready to begin the second step of protein synthesis. Translation. After exiting the nucleus, the entire mRNA strand moves into the cytoplasm. The tRNA or transfer RNA is found in the cytoplasm. It has one amino acid known as anticodon on the attachment side that correlates to the nucleotides on the other end. The term codon refers to the complementary triplet nucleotides on the mRNA strand. As the codon table illustrates, There are 64 potential mRNA codons that could represent any one of the 20 possible amino acids, either stop or start the codon. The newly formed RNA, as a ribosomal RNA, is moved into the cytoplasm and functions to transport proteins incorporating RNA into polypeptide chains. It accomplishes this by joining a tiny ribosome subunit to the cap and proceeds to start codon, usually AUG. The P and A sides of ribosome create when the large and tiny subunits of ribosome join together. The first tRNA accomplishes the P side, whereas the second tRNA enters the A side upon completion of subsequent codon. Peptide lingers along the amino acid on each tRNA attached to its side bond together tightly. Following this, the ribosome can proceed down the mRNA strand, and remaining tRNA molecule travels back to the cytoplasm to be reused. Up until the ribosome reaches stop, which instructs a release factor enter the A side and indicates the end of translation. A developing polypeptide chain is produced during this cycle following the completion of polypeptide chain and is released into the cytoplasm, where it fulfills its purpose with a cell as a protein. The remaining mRNA strand splits into solid nucleotides with recycled, signifying the end of polypeptide chain.